Text, Microsoft Teams, ODD Seminar Series, Expert Roundtable, Approaches to Translatum. 2025-06-11-1502 UTC recorded automatically.
I'm Barbara Sorkin. You'll hear in a few minutes. Lots of people needed to be admitted. Sorry about that. Good, I think we're good.
Text, Strategic Plan, 2025 to 2029, https://odds.nih.gov/NIH National Institutes of Health Office of Dietary Supplements. Videos of speakers are at the right. Bottom left text, Sorkin, Barbara, NIH ODD. Faded in the background are images of a man looking through a microscope, a woman spotting a little girl balancing with arms out, and a young woman looking up.
Welcome everyone to today's NIH Office of Dietary Supplements, or ODS seminar. I'm Barbara Sorkin. Before I introduce today's roundtable, a few brief announcements. First, our coming attraction, our final seminar for the 2024-2025 ODS seminar season, will be given by Dr. Hellen Oketch-Rabah, the director of the NIH's National Center for Complementary and Integrative Health. She'll be talking about their initiatives on whole person health and the nutrition continuum. What's going on here?
Text, Office of Dietary Supplements Seminar Series, odds.od.nih.gov, NIH National Institutes of Health Office of Dietary Supplements. A variety of pills, flowers, tomatoes, leaf. Text, ODS Event Guidelines, https://odsodni.gov about ODS Event Guidelines asks. Attendees are expected to uphold the professional and educational purposes of meetings by respecting the rights, views, privacy, safety, and dignity of all people in their speech, postings, and other actions. Keep all comments relevant to the event topic. Refrain from engaging in harassing speech or other inappropriate behavior. Posting insulting, obscene, discriminatory, or threatening comments. Disrupting the conversation, including with spam or repetitive posts. Use of AL-based or AL-assisted note-takers. If you see or experience a violation of these guidelines, please contact odds@nih.gov or send a private message to the meeting host. Participants who engage in any of these behaviors will be given a warning from event organizers, asked to stop the behavior, and be expected to stop immediately. Participants who continue to display inappropriate behavior after being given such a warning will be removed from the event.
If you are not on our seminar series list and you would like to attend Dr. Oketch-Rabah's seminar on at 11 a.m. on Wednesday, June 25th, if you'd like to be added to that seminar series list or future ODS seminars, my colleague Jasmine Hayes will have an exciting roundup for you for the 25-26 year, I'm sure. Please send us an email to that effect to the email address that we'll be adding to the chat. Or you can also see that email address on this slide.
Text, Office of Dietary Supplements Seminar Series. Any opinions expressed by ODS seminar speakers or attendees do not necessarily reflect the views of the National Institutes of Health, the Department of Health and Human Services, or the United States government. odsodni.gov. NIH National Institutes of Health Office of Dietary Supplements.
So you just saw a slide where we reminded you to please maintain professional behavior. You've all been muted, but please do watch what you post in the chat. And we will be delighted to have your questions. If you see any that seem urgent, please put them in the chat. We will try to get the speaker's attention to them. Otherwise, we may have time for some additional submitted questions at the end of the presentations and the Q&As, and we will try to take as many as we can at that point, and then just calling your attention to this important caveat. And with that, it is my pleasure to introduce today's natural product roundtable. We often at the Office of Dietary Supplements get questions from NIH applicants, NIH-funded investigators, and NIH staff about what the approaches and practical needs are for the commercial development of a dietary supplement or other natural product. People ask, what are the potential research and development pathways? What types of data, evidence of prior use, product characterization, safety data, efficacy data are needed for different development pathways. And so today we've invited some people with amazing expertise in this area who have really generously agreed to share their time and experiences and insights with us. So first we will introduce each other. My co-moderator for today's roundtable is ODS's Adam Kuzak. And I could not have done this without Adam. It was his brainchild, I think, more than mine. So Adam directs the Office of Dietary Supplements Analytical Methods and Reference Materials program, which develops resources to help ensure the chemical characterizations of dietary supplements and natural products are accurate and reliable. And I will turn this over now to Adam. And
thank you so much to join everybody. Thank you again to our panelists, which we'll introduce in a moment. And just to introduce Barbara, Dr. Sorkin co-directs the NIH Consortium Advancing Research on Botanicals and Other Natural Products, which we refer to as the Carbon Program.
Text Kuzak, Adam, NIH-ODE.
This is a joint effort with the NCCIH and NIH and other NIH institutes. It promotes research on the safety, effectiveness, and mechanisms of action of botanical dietary supplements. And we have, again, a very esteemed panel joining us today. Quickly, you can see on screen, we have Dr. Hellen Oketch-Rabah, who will speak with us first. We'll also then hear from Dr. Freddie Ann Hoffman, Dr. Amy Rowe, and then Dr. Jeff Chen. And want to be able to provide enough time for our speakers to give some opening remarks, provide us some of their insights on the topic area. So Helen, if you can please pull up your slides to share. While you're doing that, I'll give some further introduction.
Text FDA U.S. Food and Drug Administration Human Foods Program. Translating natural product dietary supplement research to improve public health. Hellen Oketch-Rabah, Ph.D. Deputy Director, Office of Dietary Supplement Programs, Office of Food Chemicals Safety Dietary Supplements and Innovation, OFCS-DSI, Human Food Program, HFP, U.S. Food and Drug Administration, NIH Expert Roundtable, June 11, 2025.
So Dr. Oketch-Rabah is the Deputy Director of the Office of Dietary Supplement Programs, ODSP, at the Human Foods Program of the Food and Drug Administration, where she oversees dietary supplements regulatory activities. From 2011 until her move to FDA in 2023, she served in a variety of roles in the Dietary Supplements and Herbal Medicines Department of the U.S. Pharmacopoeia, where she developed dietary supplements quality monographs. And she is also a member of the Botanical Safety Consortium Steering Committee. So Helen, please go ahead and kick off our meeting. Thank you.
Thank you, Adam. I hope everybody can hear me. So good morning, everyone, and thank you for that wonderful introduction, Adam. It is my pleasure to participate in this roundtable on translating natural products, dietary supplement research to improve public health. I will be giving the FDA perspective on what this would entail.
Text FDA U.S. Food and Drug Administration. FDA Organization Chart, Human Foods Program, HFP, effective October 1, 2024. A flow chart titled Department of Health and Human Services Food and Drug Administration Human Foods Program. A legend at the left shows a blue color, which the entire flow chart is, labeled, directly impacted by reorganization, that took effect on October 1, 2024, i.e. shifts of functions, resources, and/or personnel. First row, from right to left, Office of Executive Programs Project and Portfolio Management Staff 1, Project and Portfolio Management Staff 2, Legislative and Program Issue Coordination and Executive Correspondence Staff, Deputy Commissioner for Human Foods Management Operations Staff, Office of Strategic Programs. Second row left to right, Nutrition Center for Excellence, Office of Nutrition and Food Labeling, Office of Critical Foods, Office of Microbiological Food Safety, Office of Dairy and Seafood Safety, Office of Produce Safety. The following box is outlined in yellow, text, Office of Food Chemical Safety, Dietary Supplements and Innovation, Operations Staff, Innovative Foods Staff, Office of Pre-Market and Additive Safety, Office of Post-Market Assessment, Office of Dietary Supplement Programs, Office of Resource Management, Division of Budget and Planning, Division of Workforce Management, Division of Information Technology Management, Division of Safety and Facilities Management, Division of Travel Management and Operations Support, Division of Management Support and Analysis. Office of Surveillance Strategy and Risk Prioritization, Advisory, and Operations Staff, Division of Surveillance and Data Integration, Division of Risk Assessment and Prioritization, Division of Evaluation and Population Health Services, Office of Quality Assessment and Management, Quality Assessment Staff, Quality Management Staff. Third row left to right, Office of Communications, Education, and Engagement, Internal Communications Staff, Division of External Communications and Consumer Education, Division of Public Engagement and Information, Division of Web, Visual, and Virtual Communications, Office of Compliance and Enforcement, Administrative Staff, Compliance Initiatives Staff, Office of Compliance Operations and Implementation, Office of Enforcement, Office of Compliance Intervention and Consultation, Office of Integrated Food Safety System Partnerships Single Cross, Partnership Services Staff, Office of Domestic Partnerships, Office of Retail Food Protection, Office of Policy and International Engagement, Office of Policy, Regulations, and Information, Office of Policy Initiatives and Projects, Office of International Engagement, Office of Laboratory Operations and Applied Science Double Cross, Office of Regulatory Testing and Surveillance, Office of Applied Microbiology and Technology, Office of Chemistry and Toxicology, Office of Scientific Coordination and Computational Sciences, Office of Coordinated Outbreak Response, Evaluation, and Emergency Preparedness, Division of Outbreak Data Management and Signals, Division of Response, Division of Preparedness and Emergency Programs. At bottom left single cross note text, OIFSSP will work in close coordination with the Center for Veterinary Medicine. At bottom right double cross note text, OLOAS will work with the Office of the Chief Scientist and Center for Veterinary Medicine.
But first, I wanted also to give people an idea of where we are in the FDA. Last year, we had a big reorg, and we ended up with the Humans Food Program. So currently, our office is located in the yellow box there, Office of Food Chemical Safety, Dietary Supplement and Innovation, a very long name. So we no longer have CIFSAN. So just wanted to put that out there.
Text, Regulatory Framework. The Dietary Supplement Health and Education Act of 1994, designed to balance consumers' right to access to safe, high-quality, and accurately labeled dietary supplements, FDA's obligation to protect the public from unsafe and otherwise unlawful products. Image of the Act with text, S784 Act.
Okay, in developing products, dietary supplement products, as a researcher, first and foremost, if you intend to translate your research into a dietary supplement program, you need to understand the regulatory framework for what you're getting into. You need to understand the Dietary Supplement Health Education Act, that is the law that governs dietary supplement regulation. The SHARE, as it is called, was designed to preserve the consumer's access to safe, high quality, and accurately labeled dietary supplement products. FDA has the authority to protect consumers from dangerous and otherwise unlawful products. Importantly, you need to keep in mind that dietary supplements are a category of food. So food regulations apply as well as dietary supplement specific regulations. The SHARE does not generally require pre-market safety or approval of dietary supplement by the FDA. So it is your responsibility as the developer and as the manufacturer to ensure your product is safe, accurately labeled before you put it into the market.
Text, FDA, definition of a dietary supplement asterisk. Product, other than tobacco, that is intended to supplement the diet. Product that is intended for ingestion, contains one or more dietary ingredients. A. Vitamin, B. Mineral, C. Herb or other botanical, D. Amino acid, D. Dietary substance for use by man to supplement the diet by increasing the total dietary intake. F. A concentrate, metabolite, constituent, extract, or combination of any of the above dietary ingredients. Does not include certain drug ingredients. Asterisk at bottom text, see section 201, FF, of the FD&C Act for the full definition.
Now, the SHARE also defined what is a diet ingredient and a dietary supplement for the first time. You need to understand what a dietary ingredient is. For example, if you're working on a natural product which is a botanical, it would then probably fit as a diet ingredient under section 201 FF1, C, and F. In other words, maybe a botanical powder or a botanical extract for F. You'll also need to understand that a dietary supplement is intended to supplement the diet and not to be eaten as a sole meal. So whatever product you are developing must be ingested. If, for example, you are developing a sublingual product, that cannot be a supplement. It must contain at least one diet ingredient and cannot contain certain drug ingredients. And if you need additional clarification, we are always ready to point you in the right direction.
Text, dietary supplement regulatory responsibilities, firm's obligations, facility registration, new dietary ingredient notification, good manufacturing practice, dietary supplement labeling, structure function claim notification, adverse event reporting. FDA is authorized to take post-market action.
Now, since FDA doesn't preview or approve dietary supplements before going into the market, you are free to develop your supplement and introduce it into the market without engaging with the FDA at all. But you need to know your responsibilities. It is your obligation as a manufacturer to register your facility with the FDA, understand the requirement for new diet ingredient notification, follow current good manufacturing practices, ensure proper labeling of your dietary supplement, notify FDA of structure and function claims that you have on your product, and be prepared to report serious adverse events that are associated with your product 15 days after learning of the same. Now let's look at each of these in turn.
Text, FDA, facility registration. All food facilities must register with FDA domestic and foreign basic information, name, address, type of activity conducted at the facility for each food product category, responsible party FDA inspection acknowledgement renewal of information every even-numbered year.
Facility registration. Keep in mind that all food facilities must register with the FDA. This comes from the Bioterrorism Act of 2002, which was a result of the 9/11 terrorist attack in 2001. So all domestic or foreign facilities manufacturing diet supplements, holding diet supplement, must provide the information listed on the slide to the FDA. And this information needs to be renewed if there are any changes every even numbered year. So, for example, in 2024, 2026, 2028.
Text, new dietary ingredients notification asterisk, limited to certain new dietary ingredients, NDIs, not marketed in a dietary supplement prior to October 15, 1994, not otherwise in the food supply as an article used for food in a form in which the food has not been chemically altered. Manufacturers must submit a notification, NDIN, at least 75 days prior to marketing. FDA has 75 days to review and respond, acknowledgement, not approval, with no objection, generally based on deficit in identity and/or safety. Notification is made public after 90 days. Asterisk note below, Section 413, D, of the FD&C Act, 21 U.S.C. 350b, D.
And then the share also requires NDI notification to be filed with the FDA. So, determine if your ingredient is a new diet ingredient and needs a notification. So, for example, consider whether your ingredient was marketed before 1994, which would make it an old diet ingredient, but you still need to look at it carefully and determine whether you need to put in an NDI. Check if your ingredient is in the food supply in a form not altered. For example, if you're looking at a fermented broccoli extract, this one is not the same as broccoli. So even though broccoli is a well-known food and in the food supply, your fermented broccoli extract may actually be an NDI and you may need to submit an NDIN. You submit the NDIN in time so that you can go to market after 75 days. In other words, plan accordingly. Consult NDIN guidelines from the FDA, which are online. And if you wish, you can also request for a pre-NDIN meeting with the FDA. FDA has 75 days to review and respond to you. The response is usually an acknowledgement and not an approval. Response can be with no objection, so you can go to the market after that, or with objection, and the objection is usually generally based on deficit in identity or deficit in safety information. At the end, the notification will usually be made public after 90 days, but proprietary information, of course, is redacted.
Text, new dietary ingredient notification. New dietary ingredient notification must include description of the new dietary ingredient, identity and manufacturing, description of the dietary supplement, formulation, amounts, conditions of use, history of use or other evidence of safety establishing the NDI will reasonably be expected to be safe, documented history of safe use, preclinical studies, clinical studies, etc. www.fda.gov, Human Foods Program.
Now, a proper NDI notification must include the following: the name and address of the manufacturer or distributor that is introducing it into the market, ingredient identity and manufacturing information, which is very important as it relates to safety, information on the dietary supplement that contains that ingredient, that is the formulation type, the amount, and the conditions of use of that dietary supplement. And then importantly, it must include safety information that will show that that can be based either on the history of safe use or based on other evidence that supports the fact that the ingredient, when used as directed, is reasonably expected to be safe.
Text, Good Manufacturing Practices, Current Good Manufacturing Practices, CGMP, for dietary supplements, codified in 21 CFR 111 applies to all firms who manufacture, package, label, or hold dietary supplements to help ensure dietary supplement product quality, purity, consistency, and safety, production and process controls, testing requirements for raw materials and finished products.
Then the share also included provision for current good manufacturing practices, CGMPs. As a manufacturer, you are required to follow CGMPs, which is the main requirement, the minimum requirement for manufacturing, packaging, labeling, and holding of dietary supplement. Follow CGMPs to ensure the quality of your dietary supplement, to ensure that your dietary supplement is packaged, labeled, and labeled as specified in the master manufacturing records, and also build quality into your product. Start by set specifications for your raw material and finished product. Use methods that are scientifically valid and fit for purpose for your testing. Make sure the results for a product that meets your specification should meet specifications, and the finished product should meet specifications throughout its life cycle.
Text, FDA Dietary Supplement Labeling. Dietary supplements must follow food labeling requirements. 21 CFR 101. Must be labeled as a dietary supplement. Must list all ingredients. Properly formatted supplement facts. Label name, location of manufacturer, domestic contact information for submission of adverse events.
And then the share also sets specifications for labeling of dietary supplements, how dietary supplements can be labeled. So you need to understand what is required on the label of a finished product. Your product must be labeled as a dietary supplement, and the name dietary supplement sometimes is replaced with something like the name of the product and then the word supplement. For example, vitamin A supplement or something like that. It must list all ingredients in the product, including the dietary ingredients and other ingredients. You must have a supplement facts label, which is properly formatted as required. It must include the name and location of manufacturer and include domestic contact information, particularly for submission of adverse event reports.
Text, FDA, Dietary Supplement Health Claims *, Allowed Claims, Nutrient Content Claims, characterizes the level of a nutrient. Structure Function Claims, describes the role of a nutrient or dietary ingredient intended to affect the structure or function in humans, have substantiation that the claim is truthful and not misleading. Notify FDA no later than 30 days after marketing the product containing the claim. Include the FDA disclaimer language. Health Claims Qualified health claims, characterizes the relationship of any substance to reducing risk of a disease. * note at bottom. Section 403, Art. 6 of the FD&C Act.
And then you also need to understand the kind of claims that you can make on your products. You can have the kind of health claim. You can have nutrient content claim, structure function claims, health claims, and qualified health claims. And you must keep with you at all times substantiation that shows that the claim is truthful and not misleading. You must also notify FDA not later than 30 days after marketing your product containing the claim. And FDA will usually evaluate the text of the claim and respond only if there's an issue with the claim. The product label must also include the FDA disclaimer. These statements have not been evaluated by the FDA. This product is not intended to diagnose, treat, cure, or prevent any disease. Importantly, disease claims are not allowed on dietary supplements. So you cannot use words such as treat, prevent, cure, diagnose, or disease names such as cancer. These are not permitted.
Text, useful links from FDA. Information for industry on dietary supplements. FDA. How to start a food business, food facility registration, new dietary ingredient notification process, food labeling, good manufacturing practices for food, good manufacturing practices for dietary supplements, dietary supplement labeling, small entity compliance guide on structure function claims.
Now, here are some useful links that can help you in developing your product, and always you are free to reach out to the FDA. We are very open to guide you if you need, and there is an email you can reach out to us from. Thank you very much.
Text, thank you, Hellen Oketch-Rabah, Ph.D. Deputy Director, Office of Dietary Supplement Programs, U.S. Food and Drug Administration, ODSP at fdahhs.gov.
Thanks so much, Helen. Could you stop sharing your slides, please? And while Adam switches over, I will introduce our next panelist. Our next panelist will be Dr. Freddie Ann Hoffman. Dr. Hoffman is the founder and CEO of Heterogeneity LLC, a consultancy specializing in the development of complex mixtures such as botanicals, fish oils, and probiotics. Her prior work includes key research and development and regulatory roles at the National Cancer Institute, at the Food and Drug Administration Center for Biologics Evaluation and Review, and in the Office of the FDA Commissioner, and also as Senior Director for New Technology Search and Development at Pfizer Warner Lambert Consumer. So she has a wealth and breadth of experience. Freddie Ann, please go ahead.
Text, NIH Office of Dietary Supplements, June 11, 2025, Expert Roundtable, Approaches to Translating Natural Product Dietary Supplement Research to Improve Public Health, Developing a Natural Product for the U.S. Market, Freddie Ann Hoffman, MD, Heterogeneity, LLC, Washington, D.C., info@consult.com, www.heterogeneity-llc.com.
And make sure you're not muted, Freddie Ann.
Can you hear me now? Good.
Yes.
Let me repeat, thank you for inviting me this morning. I know when Dr. Sorkin had asked me to participate, your interest was to find out what to do with a natural product, that a lot of researchers are developing products and they don't know exactly which way to go. Next slide, please. So how can one regulate, or how can one market a natural product? Can we change slides? I don't know how quickly it'll do it.
Text, U.S. regulation. Natural ingredients can be regulated as foods, conventional foods, spices, food additives, dietary supplements, drugs, OTC, prescription, biologics, e.g., allergenic vaccines, cosmetics, e.g., shampoos, devices, e.g., dental alginates, poultices, adhesives. HeterogeneityLLC.com, Washington, D.C.
Okay. Natural ingredients can be regulated for the U.S. market in many different ways. In this slide, it shows that they can be foods, and that includes the conventional foods, spices, food additives, and of course, dietary supplements as defined by the law. Drugs, which includes products like fiber products, Senna, can be non-prescription or over-the-counter drugs or prescription drugs. But they can also be biologics. For example, the allergenic vaccines that are made to, say, from grass and other products. They can be, of course, cosmetics. They can be herbals added to shampoos and other products. And lastly, they can be medical devices. That includes the dental alginates, poultices, and adhesives, for example. Next slide.
Text, principles of U.S. regulation. The regulatory category of a product is determined by its route, formulation, safety, intended use.
So how do we regulate products for the United States? It's complicated, but if you boil it down, there are four major principles. Products are regulated by the route, that is the route of administration, the formulation, which is the finished product format, safety, and the intended use. And I'm going to explain these. Next slide.
skips over a slide to one titled U.S. Market Dietary Supplements.
Natural ingredients that did I miss a slide here? Let me stay on this slide.
Goes back to the skipped slide. Text. Natural ingredients developed as dietary supplements for the United States market must conform to legal requirements. Dietary Supplement Health and Education Act of 1994. DSHEA.
Natural ingredients that are developed as dietary supplements, not a problem, for the U.S. must conform to legal requirements, as Helen's already explained, and that includes the Dietary Supplement Health and Education Act of 1994, or DSHEA. Next slide.
Text. U.S. market dietary supplements, contents, must contain one or more dietary ingredients, root, must be ingested, and exert systemic activity, formulation, tablets, capsules, soft gels, gel caps, liquids or powders. Other forms, such as a bar, chew, sachet, etc., cannot be represented as a conventional food or a sole item of a meal or diet. Safety does not present a significant or unreasonable risk of illness or injury under conditions of use recommended or suggested in labeling, or under ordinary conditions of use, does not contain adulterated ingredients. Intended use to supplement the Diet, Food, Drug, and Cosmetic Act Sec. 201 meters.
This particular law spells out very specifically the contents, which you've already seen, and a dietary supplement must contain one or more dietary ingredients. The route must be ingested and exert systemic activity. And you may have seen, perhaps online or maybe on TV, there are sometimes people that are marketing patches. These cannot really be dietary supplements. Formulation is critical. This is what really defines a dietary supplement from a conventional food. Dietary supplements can be tablets, capsules, soft gels, gel caps, liquids or powders, and specific other forms, bars, chews, sachets, that are not represented to be a conventional food or the sole item of a meal or diet. Specifically, liquids cannot be beverages. Okay, there is actually some regulation on that in guidances. Safety. The safety of a dietary supplement, they cannot present a significant or unreasonable risk of illness or injury under the conditions of use recommended or suggested in the labeling, or under the ordinary conditions of use. And Dr. Rowe will explain this a little bit further. And they cannot contain adulterated ingredients, which are controlled under GMP. But I think one of the most important items is the intended use. They are intended to supplement the diet. Next slide.
Text. U.S. marketing, dietary supplements versus drugs. On the left in a red box, text, dietary supplements, ingredients must comport with DSHEA, GRAS, NDIN. Oral route, ingested by mouth and absorbed systemically. Manufacturing requirements, DS Good Manufacturing Practices, GMPs. Safety, history of safe use, for the general population, non-clinical, toxicology, safety studies, intended use, claims, nutritional content, structure or function claims, health claim, risk reduction, unqualified qualified, other, claims of well-being, nutrient deficiency, e.g. Vit C prevents scurvy. U.S. marketing, no pre-market product approval, but pre-market notifications may be required. On the right in a blue box, text, drugs, ingredients, any articles, regardless of source, intended to be used to diagnose, treat, mitigate, care, or prevent disease and that affect the structure and or function of the body. Any route, oral, sublingual, parenteral, transdermal, topical, per rectum, etc. Manufacturing requirements, current pharmaceutical GMPs, safety, required to be sale for the targeted population, as defined by the intended uses, i.e., drug label package insert, includes history of safe use and or non-clinical safety studies, as needed. Intended use, claims, disease claims, structure or function claims. U.S. marketing drug, under non-prescription drug monograph, new drug application, NDA, pre-market approval by FDA.
On the next slide, you're going to see a comparison. And this is just a very top-line overview of the differences between dietary supplements and drugs. Both are controlled under the Food, Drug, and Cosmetic Act, and for drugs, also the Public Health Service Act, should they be biologics. Ingredients for dietary supplements must comport with appropriate laws and regulations, and you've heard of these, including GRAS, generally recognized as safe, intended for use as a food, not in GRAS intended to use as a drug. Okay. They must be oral. They must meet manufacturing requirements, and these are the dietary supplement GMPs, or good manufacturing practices, which are slightly different than the food GMPs. Safety, that there's a history of safe use or non-clinical toxicology safety studies, which you will hear about. The intended use means claims. They can have content claims, structure function claims, health claims, which are risk reduction, and there are unqualified claims that the FDA feels that there is sufficient evidence to make the claim, or qualified claims. The qualified claims are those where the agency feels that there's not sufficient evidence, and so there's a qualifying statement on the claim. There are a couple other claims which we really don't spend too much time talking about, but claims of general well-being and nutrient deficiency claims. This is about the only claim that is actually technically a drug claim, but it is grandfathered in for specific nutrients that have been well defined as deficiency states, such as scurvy. For example, a claim can be made that vitamin C prevents scurvy. That's one of the few claims you can make in terms of, in quotes, a disease claim. With U.S. marketing, there is no general pre-market approval, but there are some pre-market notifications, as previously explained. Drugs, on the other hand, are any article, regardless of source, intended to be used to diagnose, treat, to mitigate, cure, or prevent disease, and that affect the structure and or function of the body. If they don't affect the structure or the function of the body, they would likely end up as a medical device. They can be any route. Oral is one of them, but they can be sublingual, they can be injection, IV, transdermal, topical, any type of route. Manufacturing requirements are current pharmaceutical GMPs. The safety, they are required to be safe for the targeted population as defined by the intended use, and intended use is described in the drug label. Read that, the package insert. And that includes a history of safe use. That is a possibility to support a drug, and more commonly, non-clinical safety studies are needed as well. The intended use, though, the actual claims are in a bucket: disease claims and structure or function claims. So there is a little bit of overlap in terms of the structure function claims, but the key point is a dietary supplement cannot diagnose, treat, mitigate, cure, or technically prevent a disease. And U.S. marketing, they can be marketed under a non-prescription drug monograph. In that particular scenario, they are generally recognized as safe, intended for use as a drug, not as a food. And or they can be a new drug, and a new drug isn't previously known to be safe and effective until proven so, and that falls under a new drug application, and those do require pre-market approval by the FDA. Next slide.
Text: U.S. marketing intended use defines regulatory route. Below is a chart titled Intended Use with an image of a head of garlic in the center and boxes around it with text: Conventional food, fresh garlic, or health claims taken with a complete diet. It may reduce your risk to heart disease. Dietary supplement, odorless garlic capsule boosts the immune system. Foods for special dietary use, enteral formula for the management of patients with heart disease. Biologic, injected garlic vaccine for the prevention of garlic allergies. Drug, any form, cures cancer, lowers cholesterol, treats hypertension. Medical device, earplugs, prevents swimmer's ear. Cosmetic, spray and body lotion rejuvenates the skin and prevents the appearance of wrinkles. Food additive, GRAS, powder added to microwavable pizza to impart flavor. From F.A. Hoffman and T. Garvey IV, Textbook of Legal Medicine, 5th ed. 2001.
So here is kind of a general summary that I've used in the past that kind of explains that if you have a natural ingredient, it can literally fall under any of the U.S. market routes. It can be for garlic, for example, fresh garlic can be taken with the diet to reduce your risk to heart disease. That is a health claim used both for foods and for dietary supplements. But a dietary supplement can maintain healthy functions, support them. Foods for special dietary uses are for the management of very specific patients, and that particular area is a complex area to talk to the FDA about or to visit their website. Biologics, I've mentioned drugs, obviously, medical devices. If you took a garlic and a clove and stuck it in your ear and said that it prevented swimmer's ear, that could be a medical device, believe it or not. All right, so next slide. The next slide is going to talk about, very briefly, for those who would like to develop their product as a drug, there is, for botanicals, but also other natural products, the Botanical Drug Development Guidance.
Text, FDA guidance on developing non-biologic natural polymolecular products as new drugs, asterisk, for the U.S. market, investigational new drug, IND, and new drug application, NDA, process, https://www.fda.gov/media/93113/download. To the right is a cover titled Botanical Drug Development Guidance for Industry with citations below.
And that can be reached through the FDA website. And this is for non-biological natural complex polymolecular products as new drugs for the U.S. market. And with that, I will stop. Thank you.
Thanks so much, Freddie Ann. Really appreciate that. Our next presenter, our next panelist will be Dr. Amy Rowe. Dr. Rowe is a principal toxicologist in product safety and regulatory affairs in the personal healthcare division at Procter & Gamble. Prior to her work at Procter & Gamble, she was a staff scientist at the Food and Drug Administration's National Center for Toxicological Research. She serves on a variety of groups developing guidelines and methods for toxicology research relevant to dietary supplements, including the Botanical Safety Consortium and the U.S. Pharmacopea's Dietary Supplement Admissions Evaluation and Labeling Expert Committee. Dr. Rowe, please go ahead.
Text, Safety Considerations for Natural Ingredients in Dietary Supplements. Amy L. Rowe, Ph.D., DABT 11 June 2025.
Okay, thank you. Thank you for the introduction, and it's my pleasure to speak to you today. In the limited time that we have this morning, I'm just going to give you a snapshot of some considerations for approaching the safety, specifically the non-clinical or preclinical safety considerations for ingredients used in dietary supplements. But hopefully we can have some good discussion after the presentations.
Text, Significant History of Use versus Traditional Use. Table below titled Table 1 Comparison between Significant History of Use and Traditional Use. On the left, text, Significant History of Use. A concept used to describe the qualified presumption of safety, where there is evidence for safety from compositional data and from experience as an ongoing part of the diet, and possibly from other relevant routes of exposure, for a number of generations in a large, genetically diverse population. Includes a scientific evaluation of the information, which should include conclusions about safe use. A description of history of use covers the use in different defined geographic areas with information on intake levels, intake patterns, years of use, preparation, handling methods, and impact on human health, as well as addressing any potential adverse effect issues. On the right, text, Traditional Use. Is based upon knowledge and experience in a population culture, but may have limited scientific documentation. Traditional use in this regard may provide information on acute toxicity, but it is unlikely to provide information on chronic toxicity and those effects that are delayed and, thus, less likely to be detected, such as cancer, developmental toxicity, including teratogenicity, and reproductive toxicity. Information from traditional use will be influenced by the general health of the particular population and the available healthcare and health monitoring facilities. Text at bottom, Rowe et al., 2018, https://doib.org/10.1002/cpt.1132.
And Dr. Hoffman introduced the safety concept and some considerations, and that fits perfectly with my next slide, where she mentioned the consideration of safe or significant history of use. And I think that's always a great place to start when thinking about the safety of a particular ingredient, such as a botanical, for example. Oftentimes our R&D colleagues will say, it's safe. It's been used in traditional Chinese medicine for 3,000 years. And that may be very true, and that's good to know. But there are other things that we have to think about, like, in the context of the proposed use relative to the traditional use, is it the same route of administration? Is it this, if it's a botanical, is it the same part of the plant? And is it the same, dose level, for example? Is it used, chronically, where its traditional use was maybe more intermittent? And then there's the demographics of the population that you're targeting. It might be, targeted towards children, for example, and that's different than the traditional use. Or is there data in women of childbearing age or who might be pregnant or breastfeeding? So that's a great initial place to start. There's some other considerations. If you'll move to the next slide, just general considerations.
Text, other sources of information to utilize. Competitor landscape, pharmacovigilance and data, utility of consumer feedback on public platforms, P&G personal healthcare.
You might look at the competitor landscape, for example, and is what the ingredient you're looking at in dozens of products on the market. And they've been on the market for decades, being used at basically the same dose level. Or, are there one or two products in the marketplace? They're maybe only available on these obscure websites. They're manufactured by companies you've never heard of before. Or perhaps you see some evidence for products being on the market, but they've been removed, but you really don't know why they've been removed from the market. So that's a consideration. And of course, when there are products in the marketplace, you can always look for pharmacovigilance data. You can look at adverse event reports in the various databases that are available to us. Kind of an emerging area in the pharmacovigilance space is considerations for how these public platforms like Amazon can actually be scraped for consumer reporting, pharmacovigilance information. And I actually like to look at products in the marketplace through Amazon. If something has 10,000 reviews, look at the negative reviews. Are they because they don't like the shape of the bottle, the color of the capsule, or is it rash, diarrhea, abdominal pain? Believe it or not, you can actually see people reporting very serious adverse events sometimes in those public platforms. So again, those are just some initial thoughts to try to identify, potential safety gaps from a very practical standpoint. So, however, oftentimes, there are gaps for various toxicological endpoints that you might have to do further testing. So if you'll move to the next slide, I always like to start by relying on my analytical colleagues and to stress the importance of solid analytical characterization of your test material.
Text, importance of analytical characterization. Botanicals are complex mixtures of phytochemical constituents. Ideal to test whole mixtures versus individual phytochemical constituents. A chart below labeled RT00 to 55.00 with 0 to 100 on the y-axis, and a line with several sharp peaks with intervals of 0 in between. The peaks are at 1.50, 1.74, 3.26, 4.53, 6.06, 15.74 which is the highest at 100, 21.23, 23.36, 24.46, 25.54, 29.32, 30.71, 31.72, 33.11.
And I also want to remind this audience, some of you might be new to complex mixtures like botanicals, but we're not talking about single chemical entities, but rather extracts that may contain dozens, if not, well over 100 individual phytochemical constituents. And so it's important, that we have good analytical characterization, and you should always start with, at least verifying or confirming your C of A. Trust but verify, right? Even if you, have solid suppliers. But the other point I want to make in terms of the complex mixtures is it's really ideal to test whole mixtures versus individual phytochemical constituents, especially if what you're putting into that capsule or solid dose form is a whole mixture. So there's several examples where I think that when individuals test just single constituents of botanicals, it can be misleading and difficult for the safety person to make decisions about the safety or moving forward with that particular botanical. So moving to the next slide, I want to use just a couple of examples that I've experienced difficulty with, and that is understanding whether or not there's a significant botanical drug interaction.
Text in vitro approach for studying botanical drug interactions. Jackson et al. 2017, https://doi.org/10.1124/dmd.117.0754008. Table on the left titled IC30 values of components in Schisandra fruit extract on various human liver microsomal P450 mediated oxidations with values of schizandrin, gamosin A, gamosin B, gamosin C, gamosin G, and gamosin N for different compounds of 1C30, CYP182, CYP2C9, CYP2C19, CYP2D6, CYP3A4 alpha, and CYP3A4 beta. On the right is a graph with incubation time on the x-axis in minutes from 0 to 300 and % of remaining parent on the y-axis from 0 to 150 showing downward slopes for all the lines for DMES 0.1%, SSE 30 micrograms per milliliter, RIF 10 micromoles, SJW 20 micrograms per milliliter, and SCE 30 micromoles per milliliter.
And, you'll see a lot of publications out there on, they're just very kind of screening level papers where they maybe test some individual constituents and they report some fairly potent CYP inhibition, for example, or maybe transporter interaction. And so from a toxicologist standpoint, in industry, what do you do with that? Do I need to label for an interaction? Do I, lower the dose? The acceptable level that's used on a daily basis, or do I not approve it at all? And this is a good example. The table on the left here, you'll see from a paper looking at a species of Schisandra where they reported some very potent P450 inhibition, looking at the isolated phytochemical constituents. But when we tested it on the right side here in the figure, in a wholesale model, a 2D plated human hepatocyte model, and using clinically relevant probe substrates to monitor intrinsic clearance, what we saw with these are two species of Schisandra here, was just a minor inhibition potential. And we also included some known inducers here, St. John's wort and a drug inducer, rifampin, just for comparison. And this is our vehicle control. So we concluded that using FDA drug-drug interaction guidance to help us interpret that level of inhibition, that it was minor and we really didn't even need to label for it. Moving to the next slide, I'll show another way that we used a similar model.
Text, in vitro approach for studying botanical drug interactions ROADed. 2019, https://doi.org/10.1089/ave.2018.0023. Below is a table titled BSE characterization with 3 columns. Boswellic acid content as 100 mg per gram. Rowe et al. 2019 BDI study. Boswell and Super with red arrows pointing to the first 2 rows showing total boswellic acids and total beta boswellic acids, and the last row circled in red 3-acetyl 11-keto beta B, AKBRA 1.7%, 35.8%. 2 graphs below. Left graph titled midazolam. Right titled diclofenac. Both graphs have incubation time on the x-axis, percent of remaining parent on the y-axis, and downward slopes of the 4 lines labeled 0.1% DMSO, 10 micrograms per milliliter BSE, 10 micromoles rifampicin, 20 micrograms per milliliter SJW, but the graph on the left has more of a spread between the lines and some dip lower than the lines that remain tighter on the right.
But in this case, it was really to compare in another way that you need your analytical partners to fully help you characterize your extracts, but it's a way that you can use these in vitro tools to quickly compare maybe the same botanical from 2 different suppliers. And this can become particularly important when you are maybe using different types of extracts, or maybe an extract that is enhanced for certain, in this case, boswellic acids, because the botanical was Boswellia serrata. And so I had previously looked at botanical drug interaction potential with a Boswellia serrata extract, but this new one that I was asked to evaluate had a significantly different boswellic acid profile. So I felt the need to go back and do the study again to determine whether or not there was any significant change in botanical drug interaction potential. And in this case, I used the clinically relevant probes, midazolam and diclofenac, and just, to shorten the story, very minimal or moderate effects in terms of drug interaction potential. So again, I think maybe I did in that case choose to label against drugs that were, had narrow therapeutic margins, but, nothing significant. Next slide.
Flowers and leaves in a bowl, jars of pill capsules. Text, advancing botanical safety through collaborative innovation. The Botanical Safety Consortium, BSC. Website, www.botanicalsafetyconsortium.org. Email, botanicalsafety@ecglobal.org.
And these in vitro approaches, or these, new approach methods, for example, are becoming more and more useful and studied in the field of toxicology. And we're starting to look at those for their utility in assessing the safety of botanicals through something called the Botanical Safety Consortium, which my friend and colleague Dr. Oketch mentioned in her presentation. And I have the privilege of being on the steering committee with Dr. Oketch for the Botanical Safety Consortium. And I see that a number of our members are also, and other steering team members are on this call today. But the Botanical Safety Consortium is a public-private consortium, so it's led by the FDA and NIEHS, and it contains or includes members from industry, academia, and other stakeholders. And you can find out more information at the website below. Next slide.
Pill bottle on its side with capsules next to it and a leaf behind them. Text, charge of the Botanical Safety Consortium to evaluate the suitability of toxicity assays for botanicals as complex mixtures. What methods can be used to better evaluate the safety of botanicals?
The purpose of the Botanical Safety Consortium is, as I alluded to, it's to look at the suitability of toxicity assays, particularly in vitro assays, and to include in silico approaches to study the safety of botanicals. Next slide.
Text, working groups. Below is a wheel made up of pentagon-shaped leaves with text in the middle, Botanical Safety Consortium. Starting from the top of the circle, the first leaf is colored dark green, medium green, and light green in horizontal stripes and labeled pharmacognosy. The next 3 leaves are light green, all under the heading analysis and labeled data analysis, chemical analysis, and IVIVE. The next 2 leaves are medium green with the heading exposure and TK, and are labeled botanical drug interactions and ADME. The next leaf directly on the bottom is split vertically between medium green and dark green and labeled dermal toxin exposure. The last 5 leaves are dark green with the heading toxicity endpoints and are labeled genotoxicity, DVPT and reprotoxicity, neurotoxicity, and cardiotoxicity.
And we are organized to study those new tools by functioning on a number of working groups. So we have working groups established based on toxicology endpoints. So everything from cardiotox, hepatotox, neurotox, to our recently formed, I would say recently in the last year, dermal tox working group. And I have the pleasure of serving with a number of experts, many of whom are here today, on the hepatotoxicity working group. But we also have working groups looking at analysis approaches as well as exposure and ADME. Next slide.
Text, hepatotoxicity working group developing screening strategies to identify botanicals with potential hepatotoxic effects, both directly and due to botanical drug interactions. To the right is a diagram of a liver with a portion magnified of a channel with round cells on the edges of the membrane, and tree-like cells labeled energy metabolism and cholestasis and cellular adaptation, e.g., hypertrophy, glycogen overaccumulation, atrophy, with arrows pointing into the channel. Some of the round cells on the edges of the membrane are magnified to show the tree-like cells with arrows going into the other cells labeled oxidative stress, membrane damage, and DNA nucleus damage.
And so I'm going to just give you a snapshot of some of the work that we're doing as part of the hepatotoxicity working group. So we're developing screening strategies to identify botanicals with the potential for hepatotoxicity. This is a very important endpoint. You know, we're seeing a lot of reports of potential hepatotoxicity that may or may not be associated with various botanicals. So this is a very important endpoint, I think, for both regulators and industry. This also involves hepatotoxicity that might be associated with botanical drug interactions.
On the top, diagram of a sphere with a broken up surface with some dark areas on it labeled 3D primary human hepatocyte spheroids, and a flask with a plant inside, and a tray next to it labeled exposure to botanical extracts. Both have an arrow pointing to bracketed items, cytochrome P453A4 inhibition induction, albumin levels in culture medium, cellular structures via cell painting, transcriptions, cell viability measured via ATP and lactate dehydrogenase. On the bottom, diagram of magnified liver cells labeled 2D primary human hepatocyte cultures, and a flask with a plant inside, and a tray next to it labeled exposure to botanical extracts with an arrow pointing to bracketed items, Metmax human hepatocyte hexcytotoxic reactive metabolite assay, cytochrome P453A4 inhibition induction, and cell viability measured via cellular ATP contents.
Next slide. covers a number of approaches that we're using. So we are using models such as the 2D primary human hepatocytes that I mentioned earlier in the botanical drug interaction studies, but also 3D primary human hepatocytes, so spheroids. And then we're looking at everything from your typical cytotoxicity assays to cellular structure effects from toxicity, using cell painting, looking at gene regulation effects, using transcriptomics, and then a number of other cell viability assays, and of course looking at botanical drug interaction potential by looking at effects on P450s. Next slide.
Text, progress and next steps, generated data in assays, additionally have data for reactive metabolites, comparison of results of extracts for LDH release, MTT, and cell painting in 2D PHH. Drafting data papers to make results publicly available. Incorporate in silico and ADM results to contextualize in vitro results. Website www.botanicalsafetyconsortium.org.
We have published the approach, that kind of our strategy for why we chose the models and the assays that we've chosen, the botanicals that we've chosen to look at, particularly for hepatotoxicity. But now we've actually run all of the botanicals through these various assays, and so we're just now kind of looking at the data, trying to understand the data, analyzing it, and trying to determine where we might have to make adjustments, some refinements. You know, does the data make sense for what we know about the botanicals that we tested, for what we know is in the literature about these botanicals? And so we're currently working on that and drafting data papers which will be publicly available. They will be in the peer-reviewed scientific literature. And in addition, we're working on incorporating in silico and ADME results to help us contextualize the in vitro results and to help us do that in vitro to in vivo extrapolation. So stay tuned for the output from these assays and our next steps. And you can always find out information about that from the Botanical Safety Consortium website. So with that, I look forward to taking your questions during the panel discussion. Thank you.
And we do have questions coming in the chat, so please keep those questions coming. And Amy, thanks for that great intro to, you know, some of the ways that we may be able, people with new dietary ingredients may not need to do as many rat studies in the future. So with that, it is my pleasure to introduce our fourth panelist, fourth and last one to present their introductory remarks, and that is Dr. Jeff Chen.
Logo of letter R in leaf shape. Text, radical science, powering proven precision wellness. Al-powered, vertically integrated decentralized trials. Jeff Chen MD MBA, co-founder CEO. Copyright 2025 Radical Science Confidential.
Dr. Chen is the co-founder and the CEO of Radical Science, a public benefit corporation that pioneered an award-winning approach for conducting large-scale, randomized, placebo-controlled trials to generate rigorous clinical evidence on the effectiveness of natural products. And his approach has significantly improved speed, affordability, and real-world translatability of these studies. Dr. Chen, please go ahead.
There we go. Can you guys hear me?
Yes, we can.
Wonderful. And you can see my
I don't
screen.
think we're seeing.
All right.
Yes, there we go.
Great. Well, wonderful. Thank you for that introduction, Barbara. And so, you know, the mission of our company, we are a public benefit corporation. It's really, what is this notion of proven precision wellness? What does that look like? How do we get there? And some of the things I'll be talking about today is, you know, uses of AI, uses of decentralization, and also this notion of how do you vertically integrate the end-to-end trial process to kind of achieve some of those efficiencies that allow you to generate large-scale real-world data or real world approximating data on these natural products.
Outline of the U.S. with bubbles of photos of diverse people all over the country. Text, radical science traction since 2021 launch.
And so before I dive into the specifics, I want to give you guys a sense of, because we've been able to harness some of these tools at Radical Science, these are some of the things that we've been able to achieve by kind of approaching clinical trials in this very new fashion.
Text, record output, blinded placebo-controlled trials on more Americans than all U.S. pharma combined.
And so we launched the company about 4 years ago, and since then we have put more Americans through placebo-controlled clinical trials than all, FDA IND drug approval trials combined. So at this point, I think we've hit nearly 80,000 Americans that we have shipped products or shipped placebos to and put them through blinded randomized controlled trials, all IRB approved, clinicaltrials.gov registered. So it's a pretty record output.
Text, generated about 100 million health data points on wellness products.
We've generated nearly 100 million health data points on these various wellness products, foods, beverages, dietary supplements.
Text, serving dozens of leading life science and consumer companies.
And we're working with some of the world's leading life science companies and CPG companies. So we have clients who are pharma companies, we have clients who are consumer companies. Everything that we do, these are non-IND studies, and we're working with foods, beverages, and supplements. So even for many of these life science companies, they have divisions focused on natural products, dietary supplements, things like that.
Text, peer-reviewed publications with academic co-authors at UCLA, Harvard, Hopkins, U of Maryland, UPitt, WashU.
Our trials have been published in peer-reviewed publications. We work a lot with academics. And so if you look at some of the papers we publish, there's co-authors from who are professors at all sorts of universities, and we love to collaborate with academics. And we now have this community of nearly 100,000 American, we call them citizen scientists.
Text, community about 100,000 American citizen scientists, more than 50% female, more than 25% racial minorities, more than 20% rural.
They're scattered all over the country, all 50 US states, and they're contributing data in our trials, and they're receiving blinded product. And they kind of have unprecedented diversity, as you guys can see, for medical research. And that's the power of a decentralized approach as well.
Text, intersection of data and medicine. Hellen Oketch-Rabah, MBA and tech CEO and entrepreneur, multiple acquisitions IPO, trustee and vice chair of UCSD Foundation, BS MBA Cornell. Dr. Jeff Chen, MD MBA and physician executive founder UCLA Natural Products Research Center, BS Cornell, MD MBA UCLA. Below are thumbnails for Female Founders Incorporated 200 2023, Top U.S. Tech Innovator KPMG, Fast Company World Changing Ideas 2023, and TED.
In terms of the makeup of the company, myself, I was a physician executive trained at UCLA in their MD MBA program, and I ran clinical trials and natural products at UCLA for a number of years after med school. And my co-founder is a serial tech entrepreneur. She's started many companies, sold them, had IPOs. Background was in applied math and predictive analytics. And we really came together to say, how do we combine the best of data and technology and medicine and science and really solve this tricky issue of taking natural products, validating their clinical effects, but also making sure that there's precision health elements. What are the right dose, the right effect, the right type of person? Can we do this all outside of the bounds of the INDs and pharma drug development, which can really is a lengthy, expensive process that doesn't really work for many of these kind of unpatentable natural products. And our company's won a lot of accolades from the business community. We were even invited to the main stage at TED, their annual conference, to give a talk.
Text, radical science proof as a service platform, scalable clinical evidence generation at 100x efficiency.
So what we kind of call this new approach, we kind of gave it a new name, because it's not simply more affordable, more rapid, more large scale, more representative trials. And so we really call this new category, proof as a service, where it's like software as a service. It's just easy. It just works. And how do you do that for clinical evidence generation? And we've created this way to scalably generate clinical evidence. It's almost about 100 times the efficiency in terms of the combination of cost and speed and scale of some of the traditional methods that I was very familiar with when I was running trials at UCLA.
Text, Radical's platform combines 1. IRB approved, blinded placebo-controlled trials.
And so we achieve this again by combining the best of technology, data, science, and medicine. So we're taking IRB approved, clinicaltrials.gov registered, double-blind randomized placebo-controlled trials.
Text, 2. decentralized, vertically integrated, tech automated.
We're taking that rigor and that protocol design, except we're doing it in 100% decentralized virtual direct consumer fashion. We also vertically integrated every single step along the way. We do everything in-house, including running our own warehouse. And the reason that's important is because you avoid all of the bottlenecks when there's tons of third parties involved. All it takes is one bottleneck in your entire clinical trial process to be delayed to delay the entire thing. But the other beauty of this is once you're decentralized, once you're vertically integrated, you can automate every step of the clinical trials process with technology, which we've done. 100% automated double-blind RCTs.
Text 3. Large diverse community of citizen scientists.
And because of this decentralization, because you've lowered the cost, you can now expand the sample size dramatically and involve all sorts of diverse individuals. So all our studies are run on Americans.
Text 4. AI optimization and analysis.
And also, with all of this, with the vertical integration, the decentralization, the automations, you now have AI manage and optimize different steps of the process. I'll talk more about that later. And now you're generating very large data sets that, yes, you can use traditional biostatistical approaches, but there's also room for AI analysis and machine learning and clustering to reveal a whole new layer of insights, because finally the data sets are large enough and diverse enough to really utilize the full power of those techniques.
Text decentralization. 1. Eliminate physical infrastructure.
So speaking of decentralization, obviously one major advantage is you eliminate physical infrastructure. You know, medical facilities are expensive. In-person clinical researchers are expensive.
Text 2. Expand participant pool by 500x.
But also, the other key thing is as soon as you eliminate the trial site, you're expanding your participant pool by orders of magnitude. It's no longer, 100,000 people that might be within striking distance of your trial site. If you're lucky and in a metropolitan area, it's now 300 million American adults who can participate. And it's also much more convenient for the participants. So one thing you'll notice in our trials, we do not pay any financial incentives to our participants. It's very convenient. They're receiving products at no cost. At the end of the study, we automatically unblind them. We send them personalized health reports. And so there's kind of this intrinsic motivation to explore products, explore their own health, get their health data back, and participate in research and be these citizen scientists that drives them.
Text 3. Reflect real world.
Because you have people that are diverse, using these products in naturalistic environments outside of artificial clinical settings, the data that you get is much more predictive of how these products are going to be used and the health effects they'll have in the real world.
Text 4. Outcomes, PROs, AI image scans, wearables, biomarker kits.
And if you think about the outcomes, yes, there's, classic validated patient-reported outcome measures, but you can also do, and I'll talk more about this, there's all sorts of interesting AI scans you can do that are clinically validated to track health parameters. There's wearables data that you can pull, and we're even shipping biomarker kits, blood, saliva, stool, vaginal testing kits, direct to consumer, self-administered, clinically validated, tested at CLIA certified labs. You can get almost all the outcomes that you can measure, or not almost all, but many of the most important outcomes you might measure in a clinical setting.
Logo, radical science. Text, vertical integration. 1. Integration of protocol design, recruitment, shipping, participant management, data management, analysis, reporting. 2. Avoid middleman delays, cost, errors. 3. Automate end-to-end.
Now, speaking very quickly about the vertical integration process, we had to do this in order to automate the entire process, in order to achieve those efficiencies and optimizations. So, we do the protocol in-house, the recruitment, the shipping from our warehouse, managing the participants, managing the data, analyzing the data, generating the reports. And really what you're avoiding is the delays, the costs, and the errors and handoffs when you have an assortment of third parties involved, oftentimes in clinical research. And also, again, only once you're vertically integrated can you automate everything end-to-end, because you control every step of the process and you get those efficiencies.
A photo of a woman, then the same photo on a phone. Both photos have text boxes over her face with measures of her heart rate, mental stress score, respiratory rate, and ANS balance. Text, AI enablement.
So before I move on to this last piece, just thinking about what are ways for those that are on the call that are conducting clinical research, what are the ways that you can leverage decentralization? What are the ways that you can bring more of the processes in-house into your own research lab, into your own company to achieve those efficiencies? And the last thing I'll talk about is some of the ways you might think about how you can leverage AI to support your trials.
Text 1. Optimize protocols and recruitment.
And so one way, because we have this large aggregate data set now on 80,000 people that we put through RCTs, the largest data set ever generated on unified clinical data set on these natural products, we can take that data set and start to better understand attrition and engagement and predicting which recruitment channels tend to result in the best screening and enrollment or tend to result in more engaged participants. You start to have a much better understanding of the nuances of the variance of some of the outcome measures you're using. That allows you to optimize various processes.
Text 2. Manage participants. 3. Analyze large diverse datasets.
We're also using AI agents, so-called chatbots, to manage participants, inquiries, safety monitor. These are large studies that we're running. Every week we're launching on average one trial, and it on average involves somewhere between 500 and 1,000 participants. That's a lot for one small startup to manage. So we had to leverage these AI agents to manage our participants. Again, we have these large data sets, machine learning, clustering analysis really comes in handy as you start to really drill down into precision health. It's not just how did men and women perform, it's how did this cluster of BMI, age, gender, severity, and caffeine usage, how does that feature set predict someone's propensity to have side effects or benefit from a product?
Text 4. Measure outcomes.
And we've been using AI to measure outcomes. So there are now facial scanning technologies, clinically validated, that can track vitals, as well as can serve as good proxies for mood, sleep, and even energy and fatigue based on pupillometry, facial microexpressions, subtle changes in facial color. And this can just be administered via participant's smartphone using their standard camera.
Text. Radical science proof as a platform. Benefits: unprecedented affordability, speed, scale, greater statistical power, likelihood of detecting true effects, multiple outcomes, multiple arms, greater diversity and generalizability, precision insights on subpopulations.
So what are some of the benefits? I mean, obviously one benefit is this is a much more rapid, affordable, and high-scale way to generate clinical evidence. But there's other things too. At larger scale, you're getting greater statistical power, increasing your likelihood of detecting true effects. You can also study, multiple primary outcomes and be powered for them, and also have many arms. So some of our trials and our next batch of trials that we're planning have as much as like 20 arms involving, thousands of Americans all at once. And so all of these multiple comparisons you can correct for when you have greater statistical power enabled by larger sample sizes. But also, most importantly, this notion of diversity, generalizability, representation. When you have large sample sizes, you don't have to be so strict on your eligibility criteria. So you can allow more diversity in, allowing your results to be more generalizable and allowing for more subpopulation analysis, that precision lens that you can take.
Graphs appear on the right for placebo, an active product with 4 lines tracked, and a red arrow pointing to the blue line with the greatest change between the two, and text. Stress improved the most for postmenopausal women.
So for example, I just pulled this from one of our recent reports that we delivered to a client where it was very clear that when you stratified the sample by female gender, and then we further dove into menstrual status, that you found that it was actually postmenopausal women that had much greater effects compared to the placebo group, compared to some of the other women in the study.
Text. Use cases. 1. R&D. 2. Precision wellness. 3. Precision marketing. 4. Claim substantiation.
All right. So how are we engaging with academia? How are we engaging with industry clients? Obviously, there's this notion of how do you use this during R&D? Could you test many formulations, products, or different doses of products all in one multi-arm trial? There's also this notion of precision wellness, precision health. What are the specific health effects of this product? And we can test multiple health outcomes all at the same time. There's also this notion of precision marketing or targeting. Who are the types of people that are most likely to benefit from this product and therefore most likely to repeat purchase and leave a good review? You can also discern that in the subpopulation analyses in our trials. And also claim substantiation. Here, I want to be very clear that it's structure function claims that I'm talking about that we're working with, which is very different than some of the other types of claims that may require different types of data or submissions to the FDA. These are structure function claims as defined by the FTC and the FDA that need to be substantiated with competent, reliable scientific evidence. And we have many clients who are using us to substantiate the structure function claims on, for instance, their dietary supplement products.
Text. Radical's transformative proof as a service approach provides gold standard clinical evidence that aligns with expectations from the FDA and FTC. Pamela Harbour, former Commissioner, Federal Trade Commission. Text. Collaboration, industry-sponsored, NIH, Foundation, SBIR, ARPA-H.
And we're collaborating with all sorts of different organizations. So, a big chunk of our work is done with industry clients, private companies, life science companies, consumer companies. But we are the named clinical research partner on all sorts of grants, foundation grants, ARPA-H grants, SBR, NIH. So if you're interested, we love collaborating. And I know a lot of the people on this call are in academia. We love collaborating. We'd love to be your research partner, and let your imagination ideas run wild.
Text, innovation engine for wellness. A mortar and pestle with leaves in it. Text, select commodity ingredients.
So I'll end with this, which is one use case of our platform. Speaking of letting your imagination run wild, you can imagine a universe where you wanted to create a product. Let's say, GLP-1s are very popular right now, but they're expensive and they have a lot of side effects. Let's say you wanted to create a natural product that could result in weight loss that maybe didn't have a lot of side effects.
An arrow points to the right to many different pill bottles and text, formulate dozens of proprietary formulations.
So you could start by selecting commodity ingredients that are already FDA approved. Maybe they're GRAS or they're already approved dietary ingredients. You can take these commodity ingredients, so to speak, and combine them into proprietary formulas and let your imagination run wild. Create dozens.
An arrow points to the right to 3 figures in text, radical trial, thousands of diverse citizen scientists. An arrow points to the right to a magnifying glass and text, analysis, millions of health data points. An arrow points to the right to figures with arrows to pill bottles and text, go to market, winner identified for each subpopulation.
And we could test all of them simultaneously in one trial against placebo in thousands of Americans, analyze the data set, and now, many of these products may fail to be significantly better than placebo, but you'll have many that will outperform placebo and have very distinct health effects in different types of individuals. And you could even test different doses of proprietary formulas. So you could have Formula 1, double dose of Formula 1, triple dose of Formula 1, Formula 2, and that's how you could stack up all these arms. And this whole process, if you sequentially did this type of development and R&D and clinical testing, it could easily take 50 plus years. No joke. And this entire process we can pull off in a matter of months.
A dotted arrow underlines the whole process with text, total duration, 6 to 12 months.
And so this is some of the type of work that we're enabling. And many of our grants that we've submitted with our academic collaborators are for these large basket studies that allow rapid R&D, innovation, precision health, and claim substantiation at the same time, because we had the rigor, the blinding for competent, reliable scientific evidence for structure function claims.
Radical science logo. Text, powering proven precision wellness, Al-powered, vertically integrated decentralized trials, Jeff Chen MD MBA, co-founder CEO, Jeff at radicalescience.com. Copyright 2025 Radical Science Confidential.
So that's my spiel today. Would love to take your questions and would love to figure out how we can collaborate. And please, if you have any questions, you want to keep in touch, please shoot me an email. And again, thank you so much for the time and the opportunity.
The speaker's videos appear.
Thanks so much, Jeff. That is something different. So excited. So the plan is that Adam and I are going to alternate voicing the questions. But before we begin with the submitted questions, and thanks to everybody, all the attendees who submitted questions, I wanted to ask whether any of the panelists have any burning questions that you would like to ask one or more of the other panelists.
Freddie Ann Hoffman raises her hand.
Not hearing any. Oh, go ahead.
I'd like to ask Jeff, how do you qualify the participants?
Yeah, oh, sorry, so I should have been clear. Yeah, so they go through screening, they go through consent. All the studies are IRB approved, they're clinicaltrials.gov registered. During the screening process, there's tons of screening questions related to either inclusion or exclusion or safety criteria. We also insert a lot of questions in the screening process that have no bearing on their eligibility, but it makes it so they can't really game the system. Our system also can flag people. It can look for people who are kind of coming back in, trying to refresh their browser history, clear their cookies, to clear the cache to come back in. But also, because we're shipping them product, we have all their addresses need to be we verify all the addresses automatically with the US Postal Service. And if we see a lot of repeat addresses, we manually review those, because we start to see these people just, trying to, there's some odd behavior here that we can then have people manually review. So it's all the standard stuff that you might have in a clinical trial. It's just, again, we've been able to do this in this decentralized, automated fashion.
And maybe if I could jump in with another question, because one of the most recent questions that came in online in the chat. How do you protect privacy of data for participants in your trials? And how do you handle informed consent? Are you HIPAA covered?
Yeah, so we comply with all the data privacy laws that are both laws as well as good practices, good clinical practice, as well as what's required by the IRB. We are not a HIPAA covered entity because there's no healthcare providers involved in this process, and we're not, pulling data from their EHR or anything like that. But otherwise, it's all standard data privacy agreements that we have, and the consent is handled via e-consent. So again, they go through screening. Our system automatically determines their eligibility. If they're eligible, they're presented with the consent. They go through it, they sign it, a copy is emailed to them, and then we have that on file as well. And so the data is confidential. And when we are reporting data back to our clients, it's all de-identified data that we're reporting back. But participants can opt in to be contacted by the funding sponsor and the manufacturers of the product after the study. And so they can kind of decide if they want to reveal their information to the other parties involved.
It's very powerful. Adam, do you want to take the next question or shall I?
Sure, thank you. And one thing I just say I failed to mention off the top, really, as we listen to these great perspectives on pathways, bringing products to getting evidence base, the one thing at NIH, there are requirements for funding, there are requirements for looking at these things, expectations, for example, of authentication of key resources or things, and glad we here at ODS we want to be a resource for people looking at that as well. And I really wanted to thank all our speakers, covered a breadth of where the types of evidence needed, implications for issues of labeling or marketing, and hearing some of those exciting cutting edge methods on toxicology and the clinical trial design. Thank you for that. A question that was submitted in advance, I think, kind of opened up to everyone on the panel. The submitter, talked about how a lot of times getting to what is the strength of the scientific evidence for effectiveness for whatever it might be, and layers of evidence in terms of thinking about bioavailability, absorption, take with food, don't take with food, the mixture of these ingredients being more effective or less effective or unknown. I think the core of the question is if the speakers can kind of share their thoughts on really verifying effectiveness for the ingredient, the formulation, what kind of evidence level would be needed either for going for something with FDA or for engaging with a decentralized clinical trial, what level of evidence or knowledge needed up front, what level of evidence needed to take a look at toxicity, et cetera. So I'll stop there. I've talked too long, but
maybe we could start with Jeff, and you could talk about your, what you've seen in terms of people using your data from your decentralized trials for health claim justification data.
Sure. Yeah, so
and then go on to some of the other panelists.
Yeah, and I'll keep it brief because there's a lot of wisdom from the other panelists as well. Again, some of the primary ways that people are using us is sometimes not even for claims specifically. Sometimes they're doing R&D. The product's not even released. There's a company that wants to make sure that the product they're releasing is actually effective and has clinically meaningful effects and is better than placebo and is better than the other formula candidates that they were experimenting with or considering, right? There are many clients who are using us with the explicit purpose of making a structure function claim on a specific health area. And about 2 years ago, the FTC revamped their entire guidance around claims on consumer health products. You may want to review that. The last time they revised it was 20 plus years ago. And they basically lay out very clearly what is competent, reliable scientific evidence by their standards for structure function claims. And it's like, generally blinded placebo-controlled trials, clinically validated endpoints, pre-registered outcomes, statistical significance, clinically meaningful, not post hoc, like et cetera, et cetera. So it's very clear that list that you can look up. So generally, we always adhere to that, and that's kind of what we our clients expect as well. But again, it's the structure functional universe that we're really big. Now, I do know that some of our clients have submitted their data from our trials in totality with other safety data. These are not necessarily safety studies. We're not doing tox studies. If we do blood biomarkers, it's not necessarily with the explicit purpose of doing PK, PD type stuff. It's usually looking at it as an outcome measure, looking at like how iron levels change, or salivary cortisol, things like that. If we're looking at biomarkers, but at least having large scale data sets from diverse people, at least showing that the reported adverse events and the severity of those reported adverse events were not significantly greater than the placebo group, that is powerful with the totality of their other toxicology data from preclinical studies, or like much smaller site-based safety studies. So we're really about effectiveness and more kind of approximating what real world safety could look like. But in isolation, our trials should not be used just for safety purposes.
Hellen, would you like to speak at all about the FDA's, your experience?
Yeah, so when it comes to evidence to support, for example, structure and function claims or nutrient claims, the evidence itself needs to be credible. In other words, if you're, for example, talking about nutrient content claim, you need to have the data that actually shows that that nutrient is in that substance, or rather in your dietary supplement in a certain amount. And importantly, whatever claim you're making, that claim should be supported by whatever the evidence that you give, and the evidence itself should be credible. And the expectation is that the data that you have, you keep this all the time. Rather, make sure you maintain that data, and if need be, make it available so that it can be verified. So in most cases, obviously, for each product, it will be evaluated on a case-by-case basis, depending on whatever claim you're making. If you're making a structure and function claim, or if you're making a nutrient content claim, you will need different sets of information.
Hellen, when people are making content claims, do you also usually see data on bioavailability of that ingredient, that component?
If you think that is what would support your nutrient content claim, then you can submit such data as well. So in other words, it's a case-by-case basis. If that is going to support your claim, then you can submit that kind of information. But most importantly, we are interested in ensuring that your claim can be supported by information that you provide, and that it is truthful. It is truthful and verifiable, if I can put it that way.
May I just add on? There actually are a couple examples that are probably very prominent in the dietary supplement realm. One of them is coenzyme Q10, and the other is turmeric and quercetin products that they really are marketing a lot, that I've observed on the bioavailability. So when you're trying to compare your bioavailability to someone else's, obviously you're going to need to show that. Whereas and I think what you said is really correct, that depends on what you're trying to claim. You better have the appropriate data to support it.
Correct.
Next question, Adam.
Sure. And this one, I'll get Dr. Rowe's opinions on seeing where there are, in the biomedical literature, many publications, a lot of times on some type of interaction, botanical or herb drug interactions, and finding something, whether it's a beneficial effect or a safety concern effect that might be like substantive in a cell model or an animal model, but then to what extent does that actually, observed in the human? To what extent does that translate? And so What are your thoughts on in terms of interpreting that data or approaches to really evaluate to trust the in vitro studies or whether predictive potential is for human effects?
Yeah, I think, a lot of the in vitro studies that are published, particularly for, drug interaction potential, you know, are are good from a screening standpoint. I think they way over predict though, in that they're using more simplistic metabolic systems. So maybe microsomes, for example, liver microsomes, so it makes it a little bit hard to jump from, microsomal studies, which are excellent studies for screening, to, clinical relevance, without doing something in between. And so what I've tried to do is come up with a strategy to do it, at least do this something in between, and then determine whether, of course, the gold standard is a clinical drug uh, or botanical drug interaction study. And so I think, I just think there's not a lot of, published studies using more physiologically relevant models. I think the other thing to be cautious about with some of the in vitro studies, um, also, is that, um, we don't have the, gut component taken into consideration. And I think for botanicals especially, that's very important to understand how they present in the gut. And so that's a missing component. There's, a lot of models, though, that are starting to be developed, that take into consideration, the gut, where you could study gut absorption, gut metabolism. So I think those are very, very exciting. But then just, sometimes just understanding the performance of your botanical, or your entire formulation, even better, in the GI tract, is also very important. So it is, how soluble is the botanical that you're using, in a relevant, gastrointestinal fluid, for example. And there are USP, of course, methods, for looking at the dissolution and disintegration of dose forms.
And there are starting to be models as well for incorporating gut microbiota into
Yeah.
that, into
Yeah,
the
absolutely.
new approach models. Helen, are you able to comment on the Food and Drug Administration's experience with submissions with new approach models? I know that FDA, like NIH, is emphasizing the shift away from vertebrate animal models, but I've also heard, is FDA seeing a lot of new approach models of non-animal models? And have you had experience with how those have been, how well those have done?
You mean the non-animal models?
Non-animal models. So in vitro, various in vitro models of the kind that Amy was talking about.
In the dietary supplement arena, we do get data, such data, but at the moment we are also evaluating the utility of these new models, and we are looking forward to being able to actually have more confidence in them before they can really apply. But definitely, information from animal models and from other NAMs are considered if they're submitted, again, on a case-by-case basis.
Adam, yours?
Sure. If I take a prerogative, there is one question in the chat that I think might be touched upon by both Freddie Ann and Helen, and it's asking if there is a blend of various herbs, botanicals, usually used in tea consumption, if those were blended together, and let's think about, would a resulting product, the question in the chat is, would it need to be qualified as a dietary supplement on the package? And would that level, a certain level of detail need to be explained in packaging materials about what's going on there? So maybe Helen, Freddie Ann, you can comment on that aspect of what needs to be, you know, done.
Sure.
You
Sure.
first, Helen.
I mean, Tisanes generally Tisanes are taken as beverages generally. However, if one wants to market a tisane as a dietary supplement, then really the intended use is what matters at the end of the day. Maybe you make a formulation with the tisane, like they said, they're making a mixture. You have to decide how you want to market your product. And as I mentioned before, a dietary supplement is not intended to be the sole meal. So a beverage cannot be a dietary supplement. So you have to decide whether you want it to be a beverage or a dietary supplement. And in some cases, you can be able to market your tisane as a dietary supplement, but it would need to be properly labeled, and it would need to be formulated in a manner that it is a dietary supplement, if I may say so. So in certain circumstances, depending on what you like to do and the formulation that you have with the tisane, it could be a dietary supplement.
Okay, I'll sort of carry on from that point. Let me just mention to this audience, I think some of you already know that my company was working with a client that actually achieved the very first a botanical new drug approval through the U.S. FDA. This is a prescription drug. The drug's name is Verigen, and it happens to come from a green tea extract. But it cannot be a dietary supplement because it's used topically for genital warts. So that's one specific use of specific extract of green tea. I should also mention that that particular extract has been used orally, and there have been some safety issues with it. I won't get into the details about that, especially a liver. So these are the things that one has to kind of look at in totality. In terms of documenting whether one can go into the FDA in a dietary ingredient setting, one has to go back and focus on the Dietary Supplement Health and Education Act of October 15th of 1994, because that split those ingredients that were on the US market before that time and those that are now coming onto the market after that time. And this is out of my expertise, but that often takes, I must say, a specialized attorney, a legal search, to go back, and it's beyond my capabilities and my pay grade, to go back and look and really document for FDA satisfactorily whether an ingredient actually meets the requirements of being a pre-October 15th ingredient, or you're going to have to go and determine through either a GRAS application or possibly through the new dietary ingredient notification. That is where you may need some actual legal help. Okay, and I'll leave it at that.
Okay, I think I have the next question, and I'm going to follow on with you, Freddie Ann, because I really want to get this question in before we run out of time. What are some of the other factors that would guide you in advising a client, whether they be a researcher or a company, in whether they have a product, they think they have a health effect? Do they go with this botanical, say, as a botanical drug, or would you advise them to sell it as a dietary supplement with perhaps a qualified or unqualified structure function claim?
There are many factors involved in that, but starting at the top and working backwards, the first step is always to imagine what is going to be on the product label, which is intended use. Okay, how are you going to use it? And again, if it's not oral and it doesn't go to the stomach and exert its effects systemically, you can cross out dietary supplement. choose something else, okay? Now, assuming that it is an oral product, then what are your other cut points? When an ingredient is being developed as a botanical drug for a specific indication, it's going to have to compete with any other drug out there on the US market or medical intervention, surgical intervention for that indication. And so basically, if you can function and you could actually get, for example, longer survival in non-small cell lung cancer, I welcome you as a botanical drug. However, if you are marginally effective, for example, for controlling diarrhea, and I challenge anybody to go to their supermarket or to their drugstore and count how many products are already out there that are generic drugs or dietary supplements or even foods like oatmeal and rice that you're going to be competing against. So a lot of what I'm going to tell you is not scientific. It is a commercial business decision. Once you get past that, you have things like safety. And we've heard about safety. Safety is another major cut point. Your product may be safe enough to treat cancer or maybe hepatitis or serious, infections, but not safe enough for your child to go to the supermarket and purchase it as a dietary supplement. And the FDA has definitely approved very, I'd say, generally unsafe products for very specific reasons. And that is a very important thing to understand. Safety for a drug is a benefit to risk assessment. That is what FDA is looking at. When the benefit for a target indication, target population outweighs the risk of using it. There is no such assessment in the non-drug realm. All right, this is very important to say. Benefit to risk is what kind of defines if you can show that your benefit for that ingredient, I don't care what it is. It can be highly toxic, like interleukin-2, which I was involved in years ago. But if it shows clinical benefit in adequate and controlled trials, all right, and that is very key. Whereas for dietary supplement, very much like a non-prescription drug, it must be pretty safe for somebody to go purchase it without the learned intermediary of a health professional, without a script. That is kind of the cut points. So it's a business decision, and it's pretty much a safety decision once you've gotten past the route and the formulation.
So I think we are running towards the end of our time, and I'm so grateful to all of our panelists for your time, your wisdom, the great comments. Before we end, there's so many great questions in the chat that we haven't gotten to. So thanks to all who attended as well and sent us questions. To any of the panelists, are there any points that we didn't cover that in our last couple of minutes you'd like to make sure people take home with them? And I'm going to start with Hellen.
Well, I just think that for those who would like to translate their work into supplements, it is really very important to understand that your supplement has to be orally administered. You have to eat it. You cannot have any other mode of administration and consider it a supplement. So keep that in mind, and keep in mind that we at the FDA are available to guide you to where you can get additional information to help you with that. We would like you to be able to do that, so we'd like to help you wherever we can by guiding you to the right information. And remember that for supplements, like mentioned before, the standard of safety is that you have to demonstrate to us in your NDIN that your ingredient is reasonably expected to be safe. It's not absolute safety, it's reasonably expected to be safe when used according to the conditions that you recommend.
Freddie Ann?
Thanks.
One last thing I'll say about dietary supplements, though, is if you're developing a product, most of the requirements are on the finished product sponsor or manufacturer. And it is the dietary supplement manufacturer that must, I don't want to say dictate, but to outline and to qualify their vendors and their source material. And that's not really something FDA's regulations have gotten involved in. And so these are specific issues, like making sure that the plant that you're using is consistent over time and verified as far as your concerns. And then the manufacturing process are inspected and are proper. That is usually through business agreements, rather than having FDA come down and saying certain things. So it's the finished dietary supplement sponsor is greatly responsible for the whole product. And that's sort of a concept to think about.
And for documenting current good manufacturing process,
Yes.
CGMP.
Vendor
Whether in the
qualification.
US or outside the US.
Wherever, yes, yeah. Now, I'm talking solely about US marketing though. Okay, I'm not speaking for products outside of the United States, because that's something I don't address.
Yeah, but I believe CGMP is required regardless where
Yes.
you manufacture.
Yeah.
Absolutely.
Yes. I mean, if it's coming here, if the end product is coming here, then the US is the prevailing jurisdiction. Yes.
Yeah. Amy?
Hi, yes, I just wanted to mention that I included a link in the chat to a paper that is, it's coming from those of us involved with the Botanical Safety Consortium, but talking about improving the rigor of conducting botanical toxicity studies with botanicals, how you go about documenting, and it has a lot of great websites. And Barbara, you were involved in that paper. Helen was involved in that paper. I see several others on the call that were authors on that paper. So I put the link in the chat, and I hope that's offers some additional help.
Yeah, I think it's going to be very exciting to see how those methods, the data from those methods aligns for chemically complex products with clinical data. And Jeff, any final take-homes for us?
I think I'm just excited about how there is a new opportunity here where, we used to rely on natural products for all our medicines for millennia. And then you kind of had this era where everything went much more kind of, synthetic and new chemical entities and all these things. And I think there's just renewed interest in this. And for the first time, you had these compounds that are, generally difficult to patent, and they're not really that interesting to pharma. But if you can somehow find this new balance between quickly figuring out their safety or quickly figuring out their actual effectiveness without having to spend $1 billion in 10 years to go through new drug development, does that offer a new kind of solution forward, especially with this growing interest in finding these modalities, non-pharmaceutical kind of non-prescription modalities. So I get really interested in that opportunity of what this looks like for health and wellness for the whole population.
Thank you all. And yes, before we say goodbye, I just want to remind everyone that the NIH has very strict requirements for the authentication of key resources, including chemically complex products. And we at the Office of Dietary Supplements, if you have questions about doing research with such products, our interest is not so much immediately in product development, but is ensuring that the research that NIH supports is replicable and that others will be able to build on it. So we have very strong and rigorous requirements for product characterization. So with that, many thanks to all of our panelists and all of our attendees for your questions and your enthusiasm. I've never seen mid-talk slides with thumbs ups and hearts before. So thanks everyone for the enthusiasm, and thank you again. We've gone over, but I'm so grateful to everyone.
They wave.
Thanks so much.
Thank you.
Thanks, everybody.
Thank you.
Thank you, Barbara. Thank you, Adam. Bye.
One by one, the videos disappear.